All, welcome to one more show. So the discussion today is very exciting, and it is by our own Dr. Paul Marik as well. So the discussion is that they have found out, Dr. Paul Marik and his team, they have found out that in the mice, so this is not a study in vivo or in humans, it is in vivo in animals. And there is a in vitro part as well. What they have found is that the S1 part of the spike protein, not the spike protein itself, the complete spike protein, but the S1 part, when combined with human ACE2 enzyme, it can generate a cytokine-like disease in mice. So for me, why this is exciting is that number one, once we know which component is causing this, we can go after that component and try to make more vaccines or more look at the drugs that can attack that part. That's one. Secondly, remember that monocyte carrying the S1 portion is also another component which probably contributes towards the long haul as well, and that is Dr. Bruce Patterson's research. So how does this all work? What did they do to find this out is exciting. I think this is the first time ever that there is an indication for what may be causing cytokine storm or what may be the pathophysiology of the cytokine storm. So let's look at that mechanism and the study itself. So here is the study. The study is published in American Physiological Society, the pathophysiology of COVID-19 and SARS-CoV-2 infection. The study is the SARS-CoV-2 spike protein subunit S1 induces COVID-19-like acute lung injury in K18H, H standing for human, ACE2 transgenic mice. Mice who have, or mice that have an ACE2 enzyme that has been humanized. So they have been their genes have been changed of these mice. So when they produce ACE2 enzyme, that is human ACE2 enzyme structure. So acute lung injury in K18H ACE2 transgenic mice and barrier function dysfunction in human endothelial part. So this part here, this is the in vitro. So there are cells that were placed in vitro, and then the S1 portion was put on them and their function was seen. So let's look at this. Here is the summary. I want to go with this summary and then I'll go to the illustrations. Check this out. This is the takeaway over here. K18H ACE2 mice that received, so this is the humanized ACE2 mice that received either saline, so that was a control group of mice, or spike protein, that was another group of mice, exhibited little or no evidence of lung injury. Wild type mice, that is non-humanized mice, and I'll explain that. I want to go over the summary first. Wild type mice that received S1 part of the spike protein exhibited a milder form of COVID-19 symptoms. So wild type means their ACE2 enzymes are not human type, they are mouse type. So when you put S1 on the mouse type ACE2 enzyme, there still is disease, but milder. Compared with K18H ACE2 mice that are the humanized. So furthermore, S1 SP, but not SP. S1 SP is spike 1 part of the spike protein, but not the spike protein itself, or all of it, decreased cultured human pulmonary microvascular transendothelial resistance and barrier function. What they're saying here is the S1 part itself can cause the barrier function to be disrupted for the endothelium or the blood vascular system. Then they're saying this is the first demonstration of a COVID-19-like response by an essential virus encoded protein by SARS-CoV-2 in vivo, and vivo being mice here. This model of COVID-19 induced acute lung injury may assist in the investigation of new therapeutic approaches for the management of COVID-19 and other coronaviruses. So that is the study's summary. I have marked the rest, but I'm going to go to our illustrations and go and explain that. So once again, welcome. Let's quickly look at the explanation. So this is the first time that cytokine-like storm is produced. And what is the basic thing over here? So if you check this diagram here, in this diagram, this is the S1 part of the spike protein. This is here is the S2 part of the spike protein. S1 part we know has a receptor binding domain, or RBD, that binds with the ACE2 enzyme, and it also has a N-terminal domain, or NTD. That's just all proteins have an N-terminal and then they have a C-terminal. Or you can say all strings, long threads, have a nitrogen side and carbon side in the case of proteins. This S1 terminal, or this S1 part, when it is separated, it is this part that causes the problem. And now we know, before I go to the study's detail, we know this thing from our previous discussions, that when S1 binds with ACE2 enzyme, we have TMPRSS2, remember? TMPRSS2, and we have other furines. These are proteins that can cleave this part. They separate the S1 from S2. That liberates the S2, that makes the S2 uncovered. And I have done this discussion in the past. And this S2 then fuses with the cell membrane. And then this S1 part goes away. And that becomes the problem. The S2 part then connects with the cell, allows the fusion of the coronavirus and the cell to occur. Messenger RNA then goes into our cell, and the remaining process of pathology occurs. So this TMPRSS2 cleaving the S1, separating it from S2, is eventually the outcome that causes then cytokine-like storm when lungs are damaged. So keep this in mind. Again, I will reiterate, there is just one sentence. If you can take away one sentence from this whole discussion, that is, first time ever, it is demonstrated in vivo, in mice, in vivo not human, in vivo mice, that cytokine storm-like condition can be produced when ACE2 with S1 part of the spike protein, they combine together. That's a very important detail. So now what did they do in their study? What they did was this. It's an animal study plus some in vitro cell study as well. The in vitro cell were human endothelial cells, plus they had transgenic mice. The mice cell had ACE2 enzyme on them that was human type. Now, we also know what they did was, the researchers, Dr. Paul Marek and his team, they also acquired S1 part of the spike protein by itself, just the S1 part, from one company. So they have given the company names. If I go in here, you would see in their materials, I'm just going to read for one, not all of them. For example, here they're saying recombinant SARS-CoV-2 spike protein subunit 1 was purchased from Ray Biotech. And similarly, they continue and explain what were the other parts in the study. So S1 parts were separately produced or purchased. The whole spike proteins were separately purchased. Then there were radioimmunoassays that were purchased to detect various components. Then they had enzymes they purchased. These enzymes can block TMPRSS2. So you can give these enzymes as sort of drugs during the test to see that if you block the TMPRSS2 and the S1 unit does not separate, what happens. So in those mice where they did not want the spike protein to become broken up into two pieces, and they wanted the whole spike protein itself to do whatever damage it can, they gave in these mice, they administered them with the protease inhibitors so that the spike protein is not cleaved and the whole spike protein stays in the mouse and does whatever damage it can. So the idea that the spike protein, whole spike protein itself, may cause damage, the research here are showing the whole spike protein, if you keep it whole, it does not cause damage or that much damage. But if you break it up into smaller pieces, then S1 part contributes to a larger damage. So for this observation, they had the protease inhibitors. They also had antibodies. Antibodies are used to bind to various parts of the molecules during the research. Then they had other chemicals as well, for example, various gels and blots and assays and kits. So these were the materials. What they then did was they prepared various mice, and there were 4 groups. What they did was, if you start here from the left side, this is one group of mice. They gave an incision on their neck to expose their trachea. The point of exposing the trachea is that when they were giving them spike proteins or S1 subunit, they wanted to make sure that they can see the catheter in the trachea of the mouse. So they exposed the neck of the mice, exposed their trachea. Then they hung the mice with their incisors. So these were anesthetized mice. These were unconscious mice. So with their teeth, they hung them, and then they injected through catheters various kind of things in their lung. For example, they injected S1 part of the spike protein in the mice that had human ACE2. Then they had the human ACE2 mice with the whole spike protein in them. They injected, not injected really, intrathecally blew it in their lungs, you can say. Then S1 spike protein in the wild type mice. That means this mouse here does not have the ACE2 modified. This mouse has mouse type ACE2. These 2 mouse or mice have human type ACE2. And then this mouse at the end also is human type ACE2, but what they injected or added to their lungs was normal saline. So these were the 4 groups. In these 4 groups, where should we keep an eye? Our eye should be on these 2 specially, then these 2 afterwards. Why is that? Here we have S1 portion, and here is another S1 portion. This group has S1 part of the spike protein, but in humanized ACE. This group has S1 part of the spike protein, but in wild type mice, non-human ACE. Then this human ACE with the spike and human ACE with the saline. These were the groups. And they have given the exact quantities of material they injected and everything. Then once the mice were introduced with those chemicals, substances, then they were kept in a warm place and they were observed on daily basis. Their bronchoalveolar lavages were done. That means what you do is you throw some water inside the lungs and then you take the water out, and you then see what kind of cytokines are present, what kind of cells are present, and you see what is inside their alveoli. So the cellular structure can be seen when you wash the lung with some fluids. So they observed the mice for clinical outcome and for the bronchoalveolar lavages. They did those 2. Then after some time, they sacrificed some of the mice and they took their lungs out and they looked at the lungs under the microscopes to see what kind of cells, what kind of damage was present. In addition to this, so this is separate. In addition to all of this, in the lab, they also acquired human endothelial cells. That is the blood vessel cells. They put them together, and these cells were, you can say they are like fake or outside the body blood vessels. They have, the cells are interacting with each other. The cells are bound to each other. They make barriers. And so you can see if the barrier function can be destroyed by something that you are experimenting with. So these cells were placed in a lab dish, and on these cells, the S1 part of the spike protein was also uh, put. And then the barrier function was observed, that how good the barrier function was maintained or not. What were the results? The results were, number one, those mice that had human ACE2 and were given S1 part of the spike protein, they very rapidly started losing weight. They started becoming sick compared to the other groups. For example, those mice that had human ACE2 but were given whole spike protein or were given saline, they almost showed no effect on their lungs. Or those mice that had wild type ACE2, mouse type ACE2, and were given S1 part of the spike protein showed only mild symptoms. Very interesting. And for the first time, the researchers showed from the bronchoalveolar lavages of the mice, they showed the cytokines that were taking part are the ones that we see in the cytokine storms. This is very important. Remember, for 18 months now, we have been thinking aloud and researching and exploring that what is the cause of the cytokine storm? Is it a person's genetic predisposition, which may still be the case? Is it a specific type of comorbidity like obesity and hypertension and diabetes and so on? But what is happening at the molecular level, at the cellular level, at the microscopic level? This is the first time they are showing that's S1 part of the spike protein. When that interacts with ACE2, human ACE2, then is the time that destruction occurs. Very interesting. They also observed that the alveoli. So alveoli are, so if we draw human lungs or any lungs, human or not, we know that the lungs have, airways, and those airways divide. And finally, there are, after many divisions, there are these tiny balloons where oxygen comes in, and then here is the blood vessel, and carbon dioxide goes out. And there is this respiratory action over there, gas exchange occur, there is respiratory membrane and so on. In here, in this alveolus, what did they find? They found, so again, those mice who were given S1 part and had human ACE2 on their cells, they saw in these mice, they saw that their alveoli were filled with fluid, there was edema. That is a sign, or that is a part of the cytokine storm. They saw septal thickening. Septal thickening is between the alveoli. Imagine if there are hundreds and thousands and millions actually of balloons sitting together. The walls of the balloons would touch each other. So where the walls are touching each other, that part is called a septum. So the septal thickening occurs. And that is a bad thing, because if the septum or the walls of the balloons thicken, then the gas exchange through that wall becomes difficult. So septal thickening occurs. Hyaline membrane is observed. What is hyaline membrane? In histology, whenever there is protein deposition in some part, under the microscope, it looks reddish, and we call it hyaline membrane. That is an indicator of damage, result, and then recovery from the damage, and the local congestion and local spilling of the proteins in that area. So it is indicator of some damage and recovery processes. Then they also saw leukocytic infiltrates. Inside the alveoli, they saw B cells and T cells and other white blood cells, which means inflammation was occurring. And then they also saw the cytokines, which were very similar to the cytokines observed in patients with the cytokine storm. So the researchers here, Dr. Paul Merrick and his team, actually created the storm in mice. This is the first time ever that this cytokine storm has been replicated in a lab. So what was the final result of all of this? What they saw was, and I'm going to repeat this, I've been saying this, S1 and S2, sorry, S1 part of spike, it's not S2, this is just spike. This is spike, S1 and S2 plus human ACE2. And here the S1 is the important part. This mouse had the most damage. If there was just a spike protein whole, not broken up, with human ACE2, no lung injury. If there was saline plus human ACE2, no lung injury. And then if there was S1 part separated in the wild type mouse, which is mouse ACE2, mild lung injury. So the takeaway, S1 part with human ACE2 is the recipe for the destruction. Let me just quickly go over some parts of this. This is the discussion for today. If you see here, I want to show you some graphs. So these are various bronchoalveolar lavage fluids. But what I want to show you, look at the markers. So they looked at interferon gamma, interleukin 1b, interleukin 6, 17, and many other tumor necrosis factors. You can see those. And here is the chart or the legend for which kind of mouse or mice showed which kind of things. And then here is the histological appearance. And if you see here, wild type down here, wild type with the S1 part of the S protein. Here in my diagrams, wherever I wrote S2, that is really spike protein, S1 and S2 together, but separated, that S1 can be cleaved off. So wild type with the S protein, this is the situation here. And if you see here, this is what is important. K18 human ACE2 plus S1 part. If you just look at this picture in general, you would see that this is more red compared to the rest, right? This seems, the top one seems the most clear. The second one is more red, and the ones after are somewhat red. More red means more inflammation is going on here, more hyalinization is happening. That means more proteins are being spilled out. That means more septal thickening is happening. And then if you look at the right side, you see a lot of dotted little purplish things. These are the WBCs over here. So this second diagram shows you a lot of damage is occurring to this lung tissue. This lung tissue almost is like non-functional. Lung tissue has to be very, very thin so the gas exchange can occur. And here, this is a good example of a thin lung tissue. And look at this, how thick this second one is. The alveoli, the balloons are almost not there. It's all just proteins and cells and inflammation and congestion, and that is what they produced. This is very similar to the septic shocks, the lungs in septic shock, plus the cytokine storm. So I thought this was a great study. Again, if I just go back, one sentence I wanted to read with you once more, and that's it. This is the first time, this green one, this is the first demonstration of a COVID-19-like response by an essential virus-encoded protein by SARS-CoV-2 in vivo. This model of COVID-19-induced acute lung injury may assist in the investigation of new therapeutic approaches for the management of COVID-19 and other coronaviruses. So this is the discussion. I hope that you liked it. For me, this was quite enlightening. Now, having said that, I'm going to hang up now. If you would like to support this work, there are links in the description to support it. I can't do chit chats today. I am collecting some documents together, and if you see my thumb here, have been putting my thumb impression or certain signatures that I've been doing. So I'll miss the chit chat today. Please remember, on this Thursday, we would have Dr. DeMello with us, and Dr. Daryl DeMello from India. So I think that will be an amazing discussion. Rima says, so is it that the breaking up of the S1 from S2 binding with S2 that is causing the problem. At least from this study, yes. Correct. Now, what are the implications for the disease? Why is it breaking off? It is breaking off. Why in some people more and others less? So all of those things can now be seen. What therapeutics are necessary? What kind of vaccine changes may be necessary, and so on. So this is a very, very important paper. Janelle says, thank you for doing a histo paper. Is this the same injury as found post-mortem on those vaxxed and unvaxxed or not? I do not know which one are we talking about, vaxxed or unvaxxed. This is mostly the in vivo on mice. I cannot correlate it. I do not know which studies you are talking about. Okay, cool. So due apologies for the chit chat, missing that today. Thank you very much for being here, and I would see you tomorrow. Please remember, on Thursday, we'll have Dr. Daryl DeMello. The time will be 9:30 a.m. in the morning. So it will not be in the evening, 6 p.m. Instead, 9:30 a.m. Pacific time in the morning. Thank you, and I'll see you tomorrow.